Welcome to solverfy.
We are being asked to determine which of the following sequences could be used to assess the risk of disease in an individual. So let's just go through all the answer choices and see what we have. Metagenomics. Metagenomics is a method to survey the kinds of species that are found in an environment. So you basically collect a sample, like let's say a soil sample or a water sample. You isolate all of the DNA from that sample. You compare that DNA and you try to determine a profile of the different species that live in that soil. So this can't really be used to tell one individual if they have a higher chance of a disease or not. It's not related to that at all. Let's look at a linkage map. A linkage map is a map of all the different genes in the genome. So it basically tells you where in the genome those genes are found. So basically every single one of these lines in each one of these chromosomes is a separate gene. And this is basically telling you just the relative positions of those genes. Note that this linkage map does not provide any information about the sequence of those genes or whether there's something wrong with those genes. So no, this could also not be used to assess the risk of disease in an individual. It doesn't give you enough information. Let's look at genomics. Genomics is a method to basically sequence all of the DNA in an organism or in an individual and essentially use that sequenced DNA to compare it to other tissues or organisms that we know are healthy or whose phenotype that we know. And in this way, you can pick out differences in potentially meaningful genes like proto-oncogenes or tumor suppressor genes and basically assess a risk of obtaining some kind of cancer or any cancer at all, or any other genetic disease. So yes, genomics is absolutely used to assess risk of disease in an individual. And choice 4 is shotgun sequencing, and this is just a method to determine the sequence of the DNA. In shotgun sequences, you are not comparing the resulting sequence to anything else to make a determination whether that stretch of DNA has anything wrong with it or not. So shotgun sequencing cannot be used for assessment of risk of disease. So the only correct choice is genomics, choice C.